Welcome to Deep Dive Dialogue on genomic products translating personalized genomics into clinical practice. Genomic products enable precise diagnosis, risk prediction, and treatment customization through tools like polygenic risk score and pharmacogenomic models. AI-driven analytics enhance accuracy, supporting real-time decisions. These innovations advance predictive, preventive, and personalized care, improving outcomes in precision medicine. To discuss this topic, we have with us Dr. S.M. Naushad. Dr. Naushad is Chief Scientific Officer at Yoda Diagnostics, Hyderabad. He has over 2 decades of expertise in biochemical genetics, human molecular genetics, and pharmacogenetics. He has held key roles at premier institutions including CDFT, NIMS, and Sastra University. Authorizing 125 high-impact papers, he has advanced research in metabolic disorders, pharmacogenomics, and epigenetics. Dr. Naushad spearheaded India's first expanded newborn screening program and developed AI-driven models for diagnosing metabolic disorders, pharmacogenomic dose prediction for 320 medications, and polygenic risk score for multifactorial diseases. His work integrates genomic, epigenomics, and metabolomics, driving innovation in precision and preventive medicine. Welcome, Dr. Naushad.
Thank you, madam.
So, Dr. Naushad, my first question to you would be, what are the most commonly used genomic products available in clinical laboratories today, and how are they shaping diagnostic and therapy of patients?
Currently, many laboratories are using whole exome sequencing, mitochondrial sequencing, clinical exome sequencing for the diagnosis of single gene disorders, and whole genome sequencing for research purpose. To identify deletions and duplications, they are using chromosomal microarray. SNP microarrays are both basically used as personalized genomic testing, predictive analytics, and other things. For cancer detection, comprehensive genomic profiling, known as CGP, is being used. And most of them target to help in early diagnosis of these disorders, mainly the genetic predisposition to these diseases, including both genetic disorders and chronic disease, and also identifying the right therapeutic module for those conditions using pharmacogenomics. Diagnosis is aimed to detect either early or asymptomatic diagnosis so that we can do lifestyle modulations through diet, physical activity, and other things, and also to understand whether how the response to therapy will be there, whether there is a possibility of any adverse drug reactions. These testings will help in minimizing those adverse drug reactions and also suggest alternative therapies available currently.
Right. So it helps us in, approaching the one size fits all doesn't suit anyone, and having a personalized treatment for every patient, and also to address the preventive. Right.
Correct.
So next question would be, how has comprehensive genomic profiling CGP testing transformed clinical practice, and what challenges remain in integrating these tests into routine care for broader population?
CGP initially will identify the single nucleotide variations, copy number variations, indels, fusions in DNA and RNA, and also provides information on tumor mutation burden, microsatellite instability, homologous recombination deficiency. So in a simple biopsy sample, we can get all the information about diagnosis, prognostic markers, and therapeutic markers. And there are 102 targeted therapies available worldwide. 4,021 clinical trials are going on. So this has transformed the oncology-based treatments effectively, increased the objective response rate, increased the overall survival, and increased progression-free survival. And many people who have undergone CGP during the diagnosis, they got benefited with this kind of approach. The challenges in India are basically affordability for the sequencing. Once you have the report, you have multiple actionable targets, choosing the right target for the therapy. And like bioinformatic pipelines, we have a variability in secondary and tertiary analysis, data interpretation, and certain therapies and clinical trials are not accessible currently to Indian population. And the tumor mutation burden rate, if you consider the prevalence is relatively low in our population. So these are the limitations currently, I can say challenges which are there in implementing these CGP tests to a broader population.
Right. But definitely, CGP is helping us in diagnostic as well as choosing the right targeted therapy. Well said, sir. So my next question
Yes.
would be that how can genomic tests reshape preventive healthcare strategies and empower individuals to take control of their health?
In India, we have high degree of consanguinity, which is increasing the risk for genetic disorders. So specifically through carrier testing, we can do a premarital counseling and prevent the burden of genetic disorders in India. So coming to chronic diseases, early detection can help in modulating our lifestyle, especially by choosing, and we can delay the onset of certain diseases through these lifestyle modulations. Genomics also will guide us in choosing what medication will suit us, what diet, what is the amount of physical activity, so that we can maintain our life in a better way without going for a strenuous activity which will affect which might result in certain cardiac arrests. So personalized medicine will has the potential to empower us in taking control of our health if we start early.
Right. So that means every individual is different, and we can address every individual with the help of genomic testing, and we can have, preventive healthcare in place. Right.
True.
So what are the key quality benchmarks for genomic testing, such as CGP, in clinical practice, and how can lab assure compliance and regulatory standards while maintaining robust analytical and clinical validity as well?
There is a consortium named Global Alliance for Genomic and Health. They have some recommendations. Like it starts with the starting material. We should have a good quality of DNA and RNA to start with. And we have many instruments like nanodrop, which will test the purity of DNA and RNA. And we have Agilent tape station. With using rinse score, we can know what is the integrity of RNA isolated. Then library preparation is the second step, where size of the library, distribution of the library, integrity of the library will play a very important role. And we can select the correct species of RNA using RNA depletion, poly A selection, so that we can target only the RNA of interest. So then QC metrics like how much yield is there, what is the read analysis, read length, GC contact, adapter content, duplication rate, Q30 statistics, error rate, cluster passing, phase and prephase percentage. These parameters will give a lot of idea about the quality of testing. And we have reference materials available through Center for Disease Control, CDC Atlanta, and National Institute of Standards and Technology. Corial samples can be used as a reference materials, and whatever the mutations identified from these samples, like the depth of the sequencing and the WAF, these parameters can be used as quality metrics for external quality assurance.
Right. And quality is the core of every analytical, test, and to give the accurate result. Right. And my last question would be, how do you envision the evolution of genetic testing over the next decade, and what emerging technologies will have the greatest impact in clinical practice especially?
With the more and more application of clinical exome, whole exome, CGP panels, the cost of sequencing is likely to come down significantly, which will help in wider adaptability and affordability of these tests at the population level. And there is a need to increase awareness among the applicability of these genomic panels, both in clinicians and public. Definitely, there is more awareness is increasing, and slowly the genetics will get integrated into routine practice. And if we have genomic data integrated with electron health records, it will help in better clinical decisions without wasting time on trial and error, and which will significantly reduce the improper diagnosis, risk for adverse drug reactions, and help in better stratification of the patients, targeted therapy, and we can have more emphasis on prevention than cure. And artificial intelligence also will help in this process through we can integrate big data analytics with the clinical data for a better AI-based clinical decision making.
Right, sir. And I believe that genomic products are really revolutionizing healthcare, how we look at a particular disease, and it is shaping a future where healthcare will be, predictive, preventive, and tailored to individual needs. Thank you so much for joining us today.
Thank you very much.